the ventrolateral mesoderm immigrate to the lateral plate mesoderm. The cells from the ventrolateral mesoderm that migrate into the adjacent parietal layer of the lateral plate mesoderm, they form infrahyoid muscle, abdominal muscles, and limb muscles. Look here in the middle of the throat, okay? You can touch a mandible or the low jaw. In the midline, we have a bones that's a hyoid, and some muscle that is between the hyoid and sternum, for example, and scapula, and we say the infrahyoid muscles. Okay, this infrahyoid muscle originate from the cells that migrate to the, and also the muscle for the abdominal wall. In the abdominal wall, we have a rectus abdominis, internal and external oblique abdominis, and transverse abdominis. We have a 4 muscle in a abdominal wall, and also the upper and lower limb muscle. All of them differentiate from the cells that immigrate to the lateral plate mesoderm. But the cell that stay here, that's the remaining cell in a myotome form, form the muscle of the back. For example, trapezius, for example, latissimus dorsi, and also shoulder girdle, for example, rhomboid, minor, major, and intercostal muscle is between we have a internal and external intercostal muscle. So the muscle in the limb come from or originate from the cells that pass through the lateral somatic frontier, this border. Then pass from it is can differentiate to the upper limb, but the muscle stay here can make the shoulder girdle, or the upper limb shoulder girdle, but the lower limb, no. All the muscle from the lower limb originate from the cell that migrate to the lateral plate mesoderm. Okay, here you can see the upper limb bud that's opposite the C3 until T2, and the muscles that migrate the cell from the ventrolateral mesoderm into the limb bud and make the muscle. Here you can see the muscle enter to the limb bud, and at first split to 2 layers. In an anterior flexor, in a posterior we have a extensor. But in the new book from I think from the 2009 organization of the muscle was different. In the new book say that we have 2 types of muscles. It's depend on the origin of the embryonic part. Some muscles that derive from the cells that emigrate to the lateral plate mesoderm. We say the abaxial muscle, abaxial region, and the muscles the dorsomedial and the cell lives here, stay here, we say the prime axial. The prime axial means near to the axis. Axis here is not occurred. Abaxial means far from the axial. So the muscle based on the region of embryonic divide to two parts: abaxial domain and prime axial domain. Prime axial domain is the dorsomedial muscle cells that from the dorsomedial myotome, and the cell myotome from ventrolateral mesoderm that stay in a before this border. Okay, stay in a paraxial mesoderm. But the cell that pass from here and migrate to the lateral plate mesoderm, they are abaxial. In the old books, the organization was, we say, the muscle epaxial, and a hypaxial. That's a depend into the innervation. The bones is correct, but we have a special difference between them, because you can see all the muscle, for example, for upper limb and lower limb are hypaxial muscle, and here this is a spinal nerve at first divide to the dorsal primary ramus and ventral primary ramus, and dorsal goes to the erectorospinal and the back muscle epaxial, and the ventral goes to the thorax muscle, the abdominal, and also the limb muscles. Hossein, do you have a question? Okay, but you can see the ventral nerves divide to the two nerves, okay? In this posterior, and here's in anterior, or ventral and dorsal. The median and ulnar originate from the anterior branch, and the radial join from come from the dorsal branch of the, for example, C5, 6, 7, 8, and T1. Okay, that's the difference between the organization of the muscle based on the embryologic origin or the innervation. As I told you, during the rotation, the dermatome change. We can see, for example, here's the C3, C5 is here, 6, 7, 8. You can check this, the little finger, and T1, and here is a T2, means the intercostal brachial nerve. It was a development of the upper limb, but something is important for the upper limb. For example, maybe you ask yourself, why the upper limb is here? The site of the location is here. Why it's not here, for example, in the middle of the belly? And the lower limb is in this part. Why is not upper? We have a tree pattern or in upper limb and lower limb that have to be know about it. At first, the positioning of the limbs is depend on the Hox gene expression. For example, in this area, we have a lot of the Hox gene, HOX1, HOX2 until about HOX13. Some of them expression in this area, and it caused to make a limb bud in this area, and it declare or detect the positioning of the upper limb. So the Hox gene can make the location of the upper limb. Then the next is a grossing of distal to proximal to distal. Okay, you know the lung of the upper limb or lower limb depend to the growth of undifferentiated zone. As I told you, in the apical part of the limb, we have apical ectodermal limb, apical ectodermal ridge that have a induction in the underneath mesenchyme. It's caused to the proliferate and don't let them to differentiate. And with this event, we have a distal growth of the limbs, upper and lower limbs, no different. And why for example, the pollex or the big toe, the pollex or thumb in a hand is in a lateral, and the little finger is in a medial. We have an area, here is this area. The cluster of the cell is near to the posterior border of the limb. You know, look here, for example, here is a okay. For example, you want to shaking hand with somebody, or the big toe here is the anterior, and here is the posterior. The cluster cell is near here. Okay, it's near here. It's for example, is limb bud, a cluster of cell. We say that's the area is a polarizing activity zone. Polarizing activity zone that express the sonic hedgehog. That's detect for you. That's the little finger is in the medial, and that's the organization of the finger is like this. The thumb in the anterior and little finger in the posterior. And after that, with the rotation, the little finger is in medial and thumb is will be in a lateral. So the ZPA is so important. Why? Because it clarified for us the organization of the finger. If we have it two, for example, a mis-expression of sonic hedgehog in an anterior part, our hand will be like this. That's a mirror image duplication of operator, because we have a two ZPA, zone of polarizing activity. And you can see it's like a mirror with a two little finger, two pointer, and two middle finger. And it's like a mirror image. It's in a lab, for example, that separate the ZPA from the chicken and transplant to the anterior border, and after that they see a mirror image of the application of limb structure like this. And they guess in a human, maybe the ZPA and the overexpression of sonic hedgehog can make a mirror image duplication of limb structure. Okay, but just leave the dorsoventral axis, means it detect for you the flexor in anterior and extensor in posterior in a upper limb, for example. In the lower limb means vice versa. Is depend on the expression of bone morphogenic protein BMP4 like this, and expression some gene can make this pattern for you. Here's show you this for the limb bud. The location detected by the Hox gene, but the outgrowing occur by the FGF. FGF is a fibroblast growth factor. Growth factor is a substance that's secret a little in a body, but it make a big changes in the body. For example, a little FGF make a growth in the upper limb. Here's the proximal occipital, and proximal distal. As I told you, we have an apical ectodermal ridge that's caused to the elongated the hand, because the cell here don't differentiation, and the cell near to the body differentiated to the bones. Anterior posterior pattern or axis, as I told you, it's depend on the ZPA that express or secret in a gene. Instead of the secretion, we say the expression, gene expression. The ZPA, zone of polarizing activity, this cell can express the sonic hedgehog, and is caused to the, for example, the organization of the hand and the foot. And for the ventral and dorsal surface of the upper limb and the lower limb depend on the some genes like a bone morphogenetic in a you can see in a dorsal surface, and when we have a bone morphogenetic here, another gene like a WNT7 expressed in a ventral. Okay. Here I show you the apical ectodermal ridge. Here's a show you FGF. FGF that's maintain the stemness cells, means an undifferentiated cell here, and far from here, that's the green part differentiated by the retinoic acid. That's a substance for the differentiation. Okay, we have some limb defect. In somebody that's absent, we have in this body have any limb that's, we say the amelia, means without any limb. The other limb is meromelia. Meromelia means a defect in the, for example, in the bone. The phocomelia is a kind of the meromelia, that's the deficiency of long bone, and you can see the finger is normal, but arm and forearm is without forearm and forearm. Micromelia means it's a hand, it's a upper limb or lower limb is like a miniature that's small and attached to the body. Is show you the brachydactyly and syndactyly. Brachy means the fingers is small. Syndactyly means fuse two fingers together, because here, as I told you, the fingers separate by the cellus, and in this hand we haven't any apoptosis between the two finger, and with the surgery it can be separated. Polydactyly means that we have an extra finger here. The extra finger is near to the little finger, but this finger is without any bones, and with the surgery it can be removed. Another defect is ectrodactyly, means the absent of the digits, for example, absent of the thumb. This one is a foot cleft. You can see the first and second finger joined together, fused together, and fifth and fourth again fused together, and third finger is absent. We say the cleft foot. That's there is a big notch between them. Okay, it's like a crap. It's like a crap. Okay. Yes, somebody maybe a membrane from the amniotic membrane separate, and it goes to the finger and make it ischemic, and you can see the fingers maybe amputated here like this. Okay, the digits amputation resulting from the amniotic band. Is this a so disaster for somebody during the gestation? For a 10 minute, a break for a 10 minute, I come back for the diaphragm and continue with the diaphragm. This is a small part. We talk about the diaphragm and the thoracic cavity. In a first week we have a fold out. We have a folding in an anterior, posterior, or cephalic, caudal, and the lateral to lateral, and make a body. But in a third week we have a embryo like this. Okay, that's a primitive streak, and the cell from here immigrate to the middle and goes to the anterior. Here is a oropharyngeal membrane, and in the anterior we have a area that's a cardiogenic area, and anterior to cardiogenic area we have a septum transversum. This cells is septum transversum. But what is the septum transversum? The septum transversum is a thick plate of the mesoderm tissue. It's between the abdominal part, here is the abdominal part, and the precardial, because we have in the lung, and that's just we have in a fourth week we have just cavities for the heart, and we say the precardial. During the folding, because the folding is like this, from its in this part goes to the inferior, and this part goes to the inferior like this from the lateral, you can see. And the septum lies here, and the cardiogenic is in anterior, okay, in this area. And the heart grows here, and after that we have a septum transversum. But this septum between the two cavity is not complete, and we have a two canal known as precardio-peritoneal canal. That's its relationship between the precardial cavity and peritoneal cavity. This septum is like a scaffold for the growing of the diaphragm on it. During the first week, from the forearm to lung, the two lung bud, it's bud, lung bud, start to growing, and that's this growing for development of the lung. And as I told you, in the anterior part here, we have a heart, but at first they grows to the posterior and fill this area, and after that grows to the ventral. But as I told you, we have heart in anterior. A new fold, this fold is a pleuroprecardial, because want to make a pleural cavity and precardial cavity. Start to the growing to the midline, and finally they separate heart from the lung bud here. And here you can see the septum transversum here and the heart during the development. And finally they join together, and in an adult we haven't any septum between the lung and the precard. Why? Because this fold means the precardio, the pleuroprecardial convert to the fibrous precardial. The fibrous precardium, around the heart we have two precards. One of them attached to the tissue, that's a visceral, and another one is fibrous, and that's a two-layer. Visceral is in the internal, in the external is a fibrous precardial. This fibrous precardial originate from this fold, pleuroprecardial membrane. So the pleuroprecardial give rise to the fibrous precardial in adult. You can see here we have a two cavity precard in the mediastinum and two lateral pleural. Okay, but as I told you, when the septum transversum is between the precardial and peritoneal, the precardio-peritoneal cavity canal is open. And after that, twofold, that's the pleuroperitoneal fold from the posterior wall, start growing until arrive to the esophagus mesentery. What's the meaning of mesentery? Mesentery is a frenulum that's a part of the connective tissue that attached to the gut to the posterior part. Finally, the pleuroperitoneal fold make a pleuroperitoneal membrane without any muscle. The muscle from the somite of the thoracic immigrate to the disc fold, and finally make a muscular part of diaphragm. At the end, the pleuroperitoneal, the blue part, cover all the septum. As I told you, this part is a septum transversum. But finally, pleuroperitoneal fold cover this area, and As I told you, the myoblasts from the lateral wall of the thorax immigrate to this membrane and make a muscular part of diaphragm, and the central tendinium in this area just made by the pleuroperitoneal fold, not transversum, septum transversum, because this fold covered the septum transversum, and septum transversum is like a scaffold, is like a base for the immigration and for the covering and pleuroperitoneal fold here. But what happened for the mesentery of the esophagus? The mesentery make a diaphragm. Wait a minute. Okay, sorry, and the mesentery of diaphragm make a cross out the diaphragm, a crura. And we have a two crura or the cross of diaphragm that was made by the mesentery. And you can see the growing of the pleuroperitoneal fold in a fourth week. In a 7 weeks, it's covered the septum transversum, and in a fourth month, the myoblasts from the lateral thoracic immigrate to the disc membrane, and finally, you can see the precardio-peritoneal canal closed completely, and as I told you, the mesentery of this gut is a esophagus, and here is a like a rope that attached to the esophagus to the dorsal surface. This is a mesentery, and this part is the septum transversum that covered by the blue part, this blue pleuroperitoneal. So the diaphragm developed from the pleuroperitoneal fold and esophagus mesentery and myoblast of the thoracic wall. But what is the rule of the septum transversum? At the first, it's about the 4 weeks during the folding of the embryo. It's lie between the show you this picture between the precord and abdominal, and that's like a scaffold for the diaphragm. In some defects, and we say the hernia, diaphragmatic hernia, and for example, you can see in the left side the here is the precardio-peritoneal is open, and we have a hernia here, and some part of the intestine come to the thoracic cavity and collapse the left lung, for example. And here you can see also the hernia. Look at the ribs of the embryo. That's not oblique. That's straight. Here, parasternal. That's a part of the diaphragmatic. We have a hernia in anterior between the sternum and diaphragm. And also we have esophagus hernia. It means a part of the stomach come into the thoracic cavity. That was the last. Yes, act as a scaffold. Thank you for Talbot. Tabassum, Fatima. That was the last slide, and it was all about the development of the musculoskeletal system, vertebra, diaphragm, the upper and lower limb. If you have a question, let me open your microphone. Have a good time. See you in exam.